Welcome to the Perception series. This video is the second part of high-level visual processing. The vision requires eye movement. Small eye movements are essential for maintaining the contrast of the objects that we are examining. Without this movement, the perception of an object rapidly fades to a field of gray. A phenomenon correlates with the decreased response of the neurons in area V1. Large eye movements direct the fovea from one object to another. These movements, or saccades, brings the high resolution of the fovea to bear on different regions of the visual field, exploring the high density of photoreceptors in the central fovea. Without saccades, this high-resolution processing could be achieved only by moving the head or body. So far in the vision videos, we discussed how visual images are constructed, beginning with the processing of intensity and contrast, then the integration of the visual primitives, and finally the high-level processing that leads to the recognition of objects. But the visual system involves more than just object recognition. It must also support the brain's goal of assigning significance to objects to develop strategies for interacting with the environment. The brain must be able to select some objects for greater examination while ignoring others. In this video, we consider how saccades support that goal. We first consider the essential benefit that saccades provide, shifting attention in the visual field and assisting with preparation grabs object. We then consider the brain mechanism that solve major problems created by saccades. The fact that the retinal image is suddenly displaced with every saccade. Now, let's shift our attention from how the brain constructs visual scene to how the brain uses visual information to plan actions. We concentrate on the region of the brain referred to as a dorsal visual pathway. This pathway extends from V1 to parietal cortex region that continues to intermediate level of visual processing such as the temporal area, and then to other parietal and frontal cortex regions. Okay, let's start with saccade. A saccade usually lasts less than 40 milliseconds and redirects the center of the sight in the visual field. Saccades occur several times per second, and each intervening period of the fixation lasts several hundred milliseconds. The image on fovea shifts with each saccade. However, we perceive a stable visual world. How come we perceive a stable visual world? One simple idea might be that the brain creates a representation of entire visual scene from a series of the visual fixation across the scene, and that what we see is this summed representation of the visual world. Well, we seem to pay attention only certain fragments of the scene. This selective visual attention relies on saccades that bring the image of desired part of the visual field onto the fovea. There are two kinds of attention have been termed involuntary and voluntary attention, or bottom-up and top-down attention. As you watch it, your attention to this video is an example of voluntary attention. If a bright light suddenly flashed, your attention would probably be pulled in involuntarily to the flash. Voluntary attention is closely linked to saccades because the fovea has a much denser array of the cones than the peripheral retina, and this permits a final analysis of object than is possible with peripheral vision. Attention, both voluntary and involuntary, has several measurable effects on human visual performance. It shortens reaction time and makes perception more sensitive. This increased sensitivity includes the ability to detect objects at a lower contrast and ignore distractors close to an object. The saccade, under voluntary control, draws attention to its goal. Clinical studies have long implicated the parietal lobe in the process of the visual attention. Patients with lesion of the right parietal lobe have normal visual fields when their visual perception is studied with a single stimulus in an uncomplicated visual world. However, when presented with a more complicated world, with objects in the right and left visual hemifield, they tend to report more of what lies in the right visual hemifield. This defect, known as neglect syndrome, arises because attention is focusing on visual hemifield ipsilateral to the lesion. Even when patients are presented with only two stimuli, one in each field, they report seeing only the stimulus in the ipsilateral hemifield. They cannot focus attention on the hemifield contralateral to the lesion, and as a result, they may not see everything in that hemifield even though the sensory pathway from the eye to the primary visual cortex is intact. This neglect of the contralateral visual hemifield extends to the neglect of the contralateral half of individual objects. Patients with right parietal defect often have difficulty reproducing drawings. When asked to draw a clock, for example, they may for all of the numbers on the right side of the face. Saccades create a major challenge for visual processing. Successive saccades produce a series of discrete images, each centered where the eye is looking. Although this result might be expected to resemble video with the camera moving about in a jumpy fashion, it does not. What neural mechanism might underline this apparent shift in images at the time of saccades? When an eye is about to make saccades, a neuron in the parietal cortex became less sensitive to stimulus already present in the receptive field and begins to respond to the stimulus that will be within the receptive field after saccades. Let's recap the receptive field concept again. There are millions of receptors in the retina, but only about 1 million optic nerve fibers send the visual signal up to the visual cortex. Consequently, individual receptors do not have a private path up to the next level of visual processing. Therefore, multiple receptors converge onto subsequent neural units on their way to the next level. This convergence results in a physiological concept known as receptive field. In simple words, a receptive field is the receptor area that, when stimulated, result in a response of the particular sensory neuron. This observation states two neural mechanisms that contribute to the stabilization of the visual scene during saccades. First, neurons shift their receptive field from one part to the visual field to another before saccades occurs. This shift in the receptive field is comparable to the change in the perceived location of the stimuli before the saccades and might contribute to the comparison in the perceived visual scene at the time of the saccades. Second, because of new receptive field position after saccades depending on the amplitude and direction of saccade, the parietal neurons must have advanced information about saccades. There are two potential sources of this information. One is the feedback to the brain from the proprioceptors in the eye muscles that could inform the brain that the eye is moving. The second potential source is the motor system that controls the movement of the eyes. This system could send a copy of its signals, known as a corollary discharge or efferent copy, to the parietal cortex to inform it that saccades is about to occur. The specific source of the corollary discharge to the parietal cortex remains unknown, but neurons in the frontal eye field that shows shift in receptive field sensitivity similar to those in the lateral interparietal area to receive an identified corollary discharge. Vision interacts with the supplementary and premotor system to prepare the hands for action. When you pick up a drink, your fingers are separate from your thumb by the width of the glass. The visual system helps to adjust this grip with it before your hand arrives at the object. Patients with parietal cortex lesions cannot adjust their grip with it or wrist angle using visual information alone, even though they can verbally describe object size or orientation. Conversely, patients with intact parietal lobe and defects in the ventral system cannot describe the size of an object or its orientation, but can adjust their grip with it and orient their hands as well as the normal subject can. The parietal lobe is a critical source of information about the visual properties of the target of the movement. How could the parietal contain this information? The representation of the space in the parietal cortex is not organized into a single map like retinotopic map in the primary visual cortex. Instead, it is divided into 4 areas that analyze the visual world in a way appropriate for individual motor system and project to areas of the premotor cortex that control individual movement. The anterior intraparietal cortex has the neuron that signal the size, depth, and orientation of the object that can be grasped. Neurons in the medial intraparietal cortex represent the target of the reaching movement and project to the frontal area, generating the premotor signal for this movement. The neural representation behind visually guided movements involve identifying targets, specifying their qualities, and ultimately generating a motor program to accomplish the movement. Neurons in the parietal cortex provide the visual information necessary for independent movement. Okay, this was the visual processing and action, and this is the last video about vision. From the next video, we will move on to the last session of the perception series, the somatosensory system. See you then.